we'll introduce recombination activating gene, RAG. This gene contributes to the diversity when recognizing antigens in the B cell and the T cell. This video is composed of 4 parts. 1. Antigen receptor. 2. Variable gene segments. 3. What is that RAG? 4. The RAG gene in the B cell and the T cell. To begin with, we'll talk about antigen receptor, which is a kind of protein recognizing antigens in the adaptive immune system. As you can see in this picture, the antigen receptor in the B cell is called immunoglobulin, and that in the T cell is called T cell receptor. Each of them has several variable regions in its structure. And by changing these regions, antigen receptors can recognize infinite number of antigens. But how does it happen? The key for this is variable gene segments. This picture shows one part of the incomplete gene encoding the variable region. This gene consists of a number of variable gene segments, which are classified into 3 groups: variable, diversity, and joining. By choosing one segment from each group and ligating them, the complete gene encoding one variable region is reconstructed. In this process, the RAG plays a very important role. Then, what is the RAG, and how does it work? As you can see in this picture, the RAG1 and the RAG2 are enzymes to cleave the before recombination gene in the recombination process, and they are encoded by 2 genes, RAG1 and RAG2. Therefore, the RAG gene is expressed only in the B cell and in the T cell. Now let's take a deeper look at the expression of the RAG gene. This picture shows how the B cell develops. As you can see, the RAG gene expresses in the early pro-B cell, the late pro-B cell, and the small pre-B cell. In the stage of the early pro-B cell, D and J of the heavy chain gene are rearranged. In the stage of the late pro-B cell, V and the complex of D and J are rearranged. In this way, the gene encoding the variable region of the heavy chain gets completed. In the stage of the small pre-B cell, V and J of the light chain are rearranged. And the complete gene encoding the variable region of the light chain is reconstructed. By the end of this stage, the recombination process by the RAG gene gets terminated, and the gene encoding the variable region is finally ready to be transcribed.